I'm going to talk about methodology transfer. I think one of the things that we've been seeing is more astronomy departments actually working closer with biology departments, which probably a few years ago might have seemed like they were as far apart as there could be in terms of how we could collaborate. But data science, in particular, I think is bringing these things much closer together. And a lot of the challenges that we begin to see that we're addressing in areas like astronomy are going on in biology as well. So there's a significant amount of leveraging and partnerships that can be developed. And so JPL has been on this path for a while. It was a curious story of me presenting a paper about what we were going to do in planetary science, about 15 years ago at the National Academy, and then getting approached by someone from the Office of the Director of the NIH that said, gee, sounds like you're describing the data challenges we're having with biology as well. Would you guys be willing to help us think about a roadmap of where we went with, we didn't call it data science then, but with a data science-like idea for areas that we're pursuing at the NIH and biomarker discovery and so forth. And so we began to look at what could we do to help them begin to really build more of a robust capability in biomarker research, and particularly cancer biomarker research. So we worked with a few institutes at the NIH and eventually formed a partnership between NASA and the NCI and JPL as part of that, and focusing on how can we build informatics data science technologies that could transfer between planetary earth science, astronomy, and address issues of reproducibility, software pipelines, being able to do more data-driven approaches, and so forth. I think the thing that I discovered after having worked in space science for many, many years is that we're very focused in space science at building these highly engineered pipelines from missions, from spacecraft, so forth. There was a lot less formality in terms of what had been happening in biology. And so at NASA, you guys know this well, we build these highly robust scientific archives. They're well curated, they're well annotated. We mandate public access after a period of time. We provide all that kind of capability. And that's a great basis for us to think about how do we shift more and more to not only being able to build these archives, but provide all the data services, as I call them, to be able to open up and provide the ability to access, analyze, and run analytical pipelines to work with the data. You may have heard some of this in Rich Doyle's talk when he talked about our full data lifecycle concept. And so as we begin to look at that, we've had this whole concept of how we go from point of collection all the way to analysis. And I'll talk about biology because it's very, very similar, actually. And so one of the capabilities I've been involved in is helping scale out the planetary data system. So that's now about 1.7 petabytes of data, 4,000 different types of data, international, so very much like IVOA, we've got the International Planetary Data Alliance, it's the ability to connect, provide a research platform is what I call it, to the international community to look at how do we break down barriers and walls of archives and start to be able to use our data and provide the appropriate data services to do that in the standards and structures. And so this is the whole data commons concept that biology and others are starting to be on a roadmap towards. And certainly one of the key things that I'll say is that I think 95% of the data that goes out of the planetary data system is imaging data. The world wants to see it. They want to analyze it. It's also the part of the archive that's certainly the largest part of the archive as well. And now you're into areas of how do we begin to run different kinds of machine learning algorithms to be able to classify, segment, and identify features in these kinds of images. And so one of the things that we developed at JPL is this scheme called MarsTrack, which loads a lot of the data from the planetary community in and has provided now tools for being able to do exploration. I always love this picture because we brought this out right when the Martian movie came out. And so the bottom right is Mark Watney. If you remember him, he was the guy who got stranded on Mars. And so the popular press used our tool to actually see if they could go to the book, track his route to find one of our old abandoned spacecraft, and things like that. And so, as we put these things together, just a lot of ability to be able to actually look at data differently, stitch together, and provide tools that can integrate that. And Mars Treks is a good example of that. There's actually a whole suite of activities now called the Solar System Treks that NASA headquarters is funding that has Vesta, and it has looking at the icy moons and certainly the moon itself and so forth. We're now on Earth. We've done water trek. We're looking at doing a methane trek, things like that, to actually open up exploration of the data. And so, as we put these things together, the NIH said, wow, we certainly would like to be able to do the similar kinds of things in biology. We'd like to be able to have robust archives of data. We want to be able to run and apply machine learning and other capabilities and algorithms to analyze that data. How could we do that in a way that we could leverage the NASA JPL expertise and some of the capabilities that you have? And I think much of it might have been thought of, okay, well, and I've had to explain this to JPL, that sometimes we can do, and George and I call this methodology transfer, where a lot of the same kinds of at least thought process can go in, but what we found is we can go a lot further. A lot of the algorithms that we're using in, say, astronomy, so Ashish has been the center of helping us do things like bringing in tools like Zooniverse for crowdsourcing, looking at how we do classification of features and data, things like that. A lot of those kind of capabilities are readily transferable between astronomy, space science, biology, and so forth. And so our mission was, how do we start to help them build what we call this biomarkers knowledge environment? Knowledge to be meaning, like, how do we capture the data, the expertise, the discovery, all the information related to cancer biomarker research. And so that's, understanding what biomarkers are themselves. So these are, indicators of disease or the potential of disease, right? So how do we find the right genes, the proteins, or those combinations of things that would say someone might be susceptible to disease? So that has always been a statistical approach to looking at how do we have enough confidence to identify those biomarkers. And so we want to identify what those are. So we want to relate those to we call biospecimens. So these are things that are taken out of humans, extracted, looked at to see if they can help us understand and identify the biomarkers that might be potential indicators. Information about various studies that we run. So we actually run and work with the NIH to help capture the data where they're looking at populations, doing recruitments, trying to understand how to identify, say, a certain biomarker like CA125. And then capturing all that study results and publications information to connect it together. And so it was interesting. Much of when I got involved, much of what they had done is they did a great job of running a routine validation study. They didn't do a good job of actually ever capturing the results to share them across centers. And so somebody had data. It was generally on their laptop or on, within their environment. They might have their own R, Python script or whatever was running in their lab, but there was no way that they were breaking down and doing joint analysis as a community of the data. And so that really is a shift in thinking to think of how do we actually do community data analysis in a way. So we are working now with two programs. One of them is called, on the left, which is what we started earlier, called the Early Detection Research Network, or EDRN. And so that's about 40 different sites focused on trying to find cancer biomarkers at the earliest stages. So imagine us being able to go and identify a marker for breast cancer, be able to have somebody take a blood test or something like that, and then be able to realize, okay, I'm at high risk. Can we detect these things earlier by looking at what markers you may have? So those might be genes, things like that could help you identify. The other program that we've been involved in, related, is something called MCL. So big word, but the molecular cellular characterization of screen-detected lesions. What that basically means is, can we identify, particularly in imaging, indicators, signatures in the data that would help us better define what cancer is? So one of the problems with cancer is in a lot of cases, the estimate here is that 60% of the time it's overdiagnosed. Radical procedure is applied. People go through all this invasive. It's not going to kill them, but the procedure might. And so how do we use statistics, machine learning, data analysis to help us classify really what are those things where you need to do the radical intervention? What are those things where you could say just watch, observe, because it's not going to kill you. So how do you begin to classify those? So what we've been putting together then is think about this as a life cycle, but the ability to capture data, metadata, run and process the data generation capabilities to be able to go from raw data to some sort of processed data, to be able to search and access that data, to be able to then combine data sets and run some sort of a joint analysis and then visualize it. So very much like the standard set of things we do in astronomy, earth science, planetary science, to be able to work with data, are things that they want to be able to do to systematize how data analysis is done. So this is just an example of starting to look at now for this MCL overdiagnosis program, bringing in the data to a data archive, a data commons, being able to capture it, being able to bring it across multiple sites. So we've got several different centers that are involved in this program as an example. And so what we're doing is helping them now build these kinds of data commons environments, plug them together, and then run various kinds of analyses, shifting them to an environment where they're not just an independent set of labs, but they're integrated together, allows us to increase the study power, provide new ways to work with the data, provide opportunities for collaboration that we wouldn't have had before. And so the NIH, and I'll show you a slide on this, is very much on this path of starting to put more and more funding and more requirements on moving the biology community into this kind of a data science kind of methodology and environment. So we're seeing a lot more investment and so forth, an opportunity for cross-collaboration. And I'm part of in addition to these, I'm part of another NCI technology program. And I recognize some of the people from our communities, people like Ian Foster and others who are also part of the same technology program. So you've seen the broadening of how the things that we've been doing in astronomy and space science or science are now being applied, and the research boundaries are being expanded, and opportunities for collaboration. So I won't spend a lot of time on so part of this is being able to access the data. But one of the things that we want to be able to do because of the characteristic of the data is create a semantic link between a lot of the kinds of data that's being generated. So if someone wants to be able to understand what biomarkers have been discovered, how do you trace that then to the publications that support it? And how do you link those publications then to the data that went into those publications and support that research? So being able to tie that all together. And so what we've done is set up an environment where you can trace all those things together. So we've got terabytes of data now coming from genomics and proteomics research that links to biomarkers. Somebody can come in and say, okay, I'm interested in CA125. What studies have been done? What publications have been produced? Where's the actual raw data? And they can go through and start to investigate all that information. And at the heart of this then is building data models and common data elements and structures and things like that, dictionaries that we're all familiar with from our space science heritage. But this community has been on the path of recognizing now that standard metadata is important. Standard metadata for being able to annotate is important. And so now we get into things like well-labeled data, training data, things like that, because we want to be able to train our data analysis is important. And so what we had to do with them was really start at the heart of figuring out what is it you're trying to annotate? What are the features? What is an image? What's a lung image? How do you be able to describe that in ways? What was the instrument that produced it? All those kinds of things that we've been looking at are now critical. And in many cases, what we were given in the beginning was, oh yeah, here's a raw image. And we had to figure out, what do we do with this raw image? What does it look like? How do we begin to describe it? And so we've been working with them to build this whole kind of data model up to support that. And the other thing that we started building then was based on this was an open source software tool we call LabCAS. It's based roughly on some work that I and others had done at JPL to build an open source data science framework called Apache ODT. It's part of the Apache Software Foundation. And LabCAS is really meant to figure out how do we bring the laboratories into the whole data ecosystem, give them the tools they need to be able to capture the data in a reliable way, to be able to insert metadata, be able to run the processing, so forth, where we could do it in a systematic manner that we can make sure that the data becomes part of this broader ecosystem. And so, and then also being able to plug in their analytical methods. And so we've been working on putting this together and getting these programs to move from doing independent analysis to integrated analysis across centers using LabCAS. And so the process is very similar to things that we would see, which is being able to register your data set and assign metadata and be able to manage that and so forth, but also being able to then launch various workflows as part of putting this in an archive, things that we do in other fields as well, to be able to take the raw data, capture intermediate results, be able to run and produce the derived products, be able to capture those, and then support the analysis. What we see a lot of times is all of those pieces are disconnected, and we're trying to put them together so that you've got all of that in one place, as well as you can reuse the analytical processes across sites so that there's consistency in how jobs are being run. And so this is just another view of that idea of being able to do it, but taking things like RNA sequencing, various kinds of imaging or pathology images, and being able to run them into this repository and then capture that and make that available and tie it together so that they want to do it. One of the big things that they can't do that they'd like to be able to do is really to interrelate data together. So let's say they have a particular specimen that was extracted from a particular human. What they'd like to be able to do is link the sequencing data and the imaging data together with some of the clinical data and being able to put that all into an integrated environment, because it's the same challenge we've got, which is you're looking at an object and we have all these instruments that took it, and then if we do independent analysis, we don't learn as much as we do if we can actually do joint analysis by putting it together. And so the way that we've been helping construct this environment is building this on AWS, and so we're shifting them to the cloud like is the popular thing to do today, using a number of open source tools. And what's interesting about this is part of the challenge is that a lot of the sequencing and it was run on local HPC environments, things like that. We can now do it in AWS for them as a service, and rather than them running these things independently, we can have a integrated algorithm for the program that somebody runs that can scale. And so we've done a number of tests on AWS to look at how we do that, throwing different servers at it, being able to use and scale horizontally. And so you can see us being able to bring it down to some point where you begin to throw enough processing at it that it's just going to flatline. But the point is that we can get to a place where we can go from days to an hour or two to be able to run things that were taking days. And so depending on the needs of the program, you can actually scale up that way, but provide as a common service. So just some examples of where we're applying this. So we've been doing a project with Dartmouth University. We moved 10 terabytes of data using grid FTP over. We ran it through LabCAS. We reproduced the results with about 99% accuracy. And we're able to demonstrate the idea of a central analytics pipeline. Another one that we're doing, and Ashish is involved in this as well, is being able to run something with Stanford and a few other partners that's using something called Smart3Seq, which is a method for quantifying gene expressions in small RNA samples. It's for single-cell science, and what's happening is that one of their programs, the MCL program, is using this methodology for one of its validation studies. Rather than every lab trying to run and do the analysis, they can upload it and we can run it, then it becomes more consistent across sites. DNA sequencing is another one. So we're doing some work with UCSD and others for looking at how to run the DNA sequencing pipelines. So that's running as well. And then one of the big investments we're doing right now is multidimensional imaging and other kinds of data. And so that has really great overlap in terms of what astronomy is trying to work on. And so high-dimensional data sets, looking at how you reduce that, looking at how you visualize that, and so forth. And so we've been working with a number of different investigators on trying to help them get their data captured, understand their data, look at it, visualize it, so forth. And part of that's been building this image archive to look at how do we capture all this imaging data in a way that they can go through it. What's interesting is that that many of a lot of the imaging data has been buried away in various medical systems. A lot of times, the instrument providers, so they get these MRI instruments or microscope and things like that that generate these images. The software that they use to analyze has almost been a byproduct. The imaging companies provide it, and they say, oh, yeah, you got to use our software. And by the way, it only works with our software, but we're trying to do these large national studies. How do we get the data out of these environments? And so we've been working to actually pull in, support the annotation, extract out what we can to create dynamic metadata and put this together into larger archives where not only they can access this collectively, because they can't do it right now, but also run various algorithms to look at and explore the data as well. And so this is something that we're starting to build onto the cloud as well. And so setting up this environment, we're creating a way to plug in different analytical tools. And so one of the conclusions we made was that we would focus on building the data and computational capabilities, working on helping them transfer methodology, but that we wouldn't build any kind of analysis, visualization tools, things like that, because there's a lot of investments by the community, and can we plug those in? And so we've been doing that. Surprisingly, a lot of our investigators are using tools that are dependent on running locally on their laptops. So the paradigm is either I'm sitting in my research lab and I've got all the data there, or I'm downloading all the data and then I'm doing my local analysis. However, as we're approaching terabyte. So a lot of this data are many gigabytes each type of data because they're multidimensional and so there's many slices. And so the challenges of downloading all that data is just certainly bandwidth and other things like that. And so we're shifting and working to figure out how to resolve that with them by pushing more of the analysis on the cloud. Other things is plugging in various viewers that exist. Many of them, again, are local to people's laptops, so trying to address that, as well as look at other approaches for doing multi-dimensional imaging, so things like VR and so forth. One of the ways that we're actually addressing this is by now pushing our data on the cloud, we're running a capability from Amazon called Amazon Workspaces. What we can do is basically virtualize create a virtual machine, sit that on top of our cloud infrastructure, provide them a desktop that's pre-configured with all the tools they need, so some of the tools they're used to, connect that in, and then they've got the data computation tools all integrated together. And this is actually pretty amazing, working really well. So there's a couple of tools that we've actually brought over. QPath, which is looking at pathology data, and so we've got a person validating that up at UC Davis. And then something called 3D Slicer, which helps work with more radiology data. And that's we've got a group at University of Miami that's working on that. And in both cases, we're actually able to go through and explore that and look at the data locally. So just examples of running some of this locally, being able to look at the data, run their tools, so forth. So they can do joint analysis, which is an annotation, which is something they couldn't do. They were calling up and running a WebEx type session, and somebody was popping up the images, and then somebody was typing in what the agreed annotations are. Now we're trying to crowdsource and collaborate how you tie it together so you actually do analysis, and you could do it independently. You don't need to be sitting there at the same time. People could do it, at 10 o'clock at night, which is a lot of time when we get our opportunity to do work. So, people get on and look at and do their analysis that way. Looking at other kinds of data, 3D Slicer, again, this is radiology data, and so forth. Now, another activity we've been involved in Ashish is at the center of this is looking at how we help them do classification of images. So they want to be able to identify what a nodule is, be able to identify features of that nodule, be able to create classifications of that. And so we've got a classification system from them that they're trying to work on. And so there's a study that's basically trying to standardize how radiologists go through and classify these features. And so we're working with them to actually take and adapt Zooniverse as a tool where they can begin to bring in some of their radiologists, go through these images, and then we can see how well they do, how consistent they are in terms of classifying the images, and see if we can start to then also build a classification model for that. So that eventually drives us towards looking at things like machine learning, if we can begin to provide a model for that. So what's been emerging from this now is now a platform similar to what we do in planetary astronomy, so forth, to be able to bring that data together for now cancer biomarker research. And so the data is being integrated, being plugged together. We've got about 1,000 biomarkers captured now, got 200 various studies going on, 1,500 publications that are connected, about 100 terabytes of data, tools that are plugged together, so forth. I heard this week that the Beau Biden cancer moonshot was an effort that Congress started this year. NIH is funding that as a big initiative for doing research. And what came up at the meeting was, are you guys all going to work with JPL in terms of how to plug this together? And so I think there's growing interest in terms of thinking about this paradigm of doing analysis versus what's been going on historically. The NIH has a data science strategy that very much follows what we're talking about, building data infrastructure, modernizing the ecosystem, the repository infrastructures, driving more to analytical tools, and so forth. And like all of us, they're worried about workforce development, but I thought this group would appreciate this quote. By 2025, the total amount of genomics data alone is expected to equal or exceed the totals from 3 other major producers of large amounts of data: astronomy, YouTube, and Twitter. My kids are a lot on YouTube, so I know how much data is there. But that's an amazing statement if that's really true. That's going to be a game changer that is going to put pressure on how they adopt more and more data science capabilities. And so our future direction is to help them continue to systematize, do more and more integration between our science areas. So that's something that I'm quite interested in helping them unify their enterprises so they've got much more of a joint collaborative approach to how they're doing data analysis and they're systematizing that. So really an opportunity to look at how we can do that. So with that, I'll take any questions. Thank you.